Welcome, NutritionRadio.org listeners. This is Lonnie Lowry, a university nutrition professor of over 20 years and podcast host of long-running shows like Iron Radio. Come on in.
Trends in Science.
Hey, everybody. You know, we're often told to avoid certain dietary components, fructose or sodium or preservatives. And as we explore today's topic, we may be adding another one to the list. There's a new paper that came out that, again, sent me down a little rabbit hole about advanced glycation end products. Some of you may have heard of these AGEs. They're typically considered bad for you. They're both in foods, especially created by dry heat, by dry cooking, but also created in the body in response to different things that we ingest. The problem is there's this sort of paradox and controversy, and people don't always agree about the details. Let's start with the name and the title of this first journal. Brand spanking new. This paper was just accepted in March of this year and just released. This is by Ashner and colleagues. It's primarily a Russian paper, Role of Gut Microbiota in the Modulation of the Health Effects of Advanced Glycation End Products. So this is a review paper. It says the existing data demonstrate that dietary AGEs, so think about them as sort of gummed up proteins or lipids and proteins, that dietary AGEs can have a significant impact on the richness and diversity of the gut microbiota. That seems to be a mechanism underlying everything these days, doesn't it? It says it's dependent on the type, the species of gut microbiota we're talking about, the exposure dose, but it also says that the emphasis here is that there's sort of a two-way street. It says, in addition, gut microbiota, not only reflecting the diet, but they can metabolize certain AGEs that we consume or are created in the gut. So that's a consideration as well. Here in the intro it says advanced glycation end products. So again, think gummed up proteins are a highly heterogeneous group of chemical species formed through non-enzymatic reactions of carbs with proteins. A big part of this is, for example, a contributor is the Maillard reaction. The Maillard reaction is something you could consider as like a browning of a food, and it can create some of these AGEs. There's just a wide variety. There's carboxymethyllysine, or CML. There's pentosidine, which is one that actually fluoresces. And I've done some research with this in the past where you can actually put a device against your forearm and see how fluorescent the skin is, because this pentosidine and other AGEs are fluorescent in nature. It's amazing to think that if you're really gummed up, you sort of glow, at least under the right light. Now let's look at the internally produced versus the external dietary ones. Internally, or endogenously formed AGEs, they're generated at high amounts in diabetes, right? Diabetes mellitus because of insulin-resistant and persistent high blood sugar. They impart toxic effects on cells because they induce oxidative stress, mitochondria dysfunction, inflammation. They've been shown to be connected with things like neurodegeneration and cancer and infertility or just aging. Now, dietary intake of these things continues to be controversial and teased apart. Dietary AGEs contribute to AGE accumulation and toxicity in the body. So these are typically highly processed, heat-treated foods, like I said, dry heat. It's been proposed that the modulation of the gut microbiota significantly contributes to the effect of these AGEs on health. So again, there's this linchpin in the middle with the gut microbiota playing a role. But let's quickly take a look. Here's the rabbit hole of AGEs in the diet. If we look at a 2010 paper here by Uri Bari and colleagues, this is Journal American Dietetic Association in 2010, Advanced Glycation End Products in Foods and a Practical Guide to Their Reduction in the Diet. So again, looking at them as something that's undesirable, trying to reduce them. It does point out dry heat promotes new dietary AGE formation by 10 to 100-fold above the uncooked state. They go on to say animal-derived foods that are high in fat and protein are generally AGE-rich. In contrast, carb-rich foods like vegetables, fruits, whole grains contain relatively few AGEs after cooking. Now, this is where some of the controversy happens, because if you look at a later paper here from 2017, they don't agree, really. The author here, De Christopher, this is Advances in Nutrition 2017, Perspective. The paradox in dietary advanced glycation end products research, the source of the serum and urinary AGEs is the intestines, not the food. This paper points out that past research has suggested that Western-style fast foods, including grilled and broiled meats, contain high levels of pro-inflammatory AGEs. However, recent studies with state-of-the-art ultra-performance methods have found that there is no evidence that these foods have elevated levels of dietary AGEs relative to other foods. Paradoxically, observational research has found that the intake of fruits, mainly apples, fruit juices, vegetables, nuts, soy, a lot of these foods that are synonymous with healthy eating are actually worse in some way because they are sources of fructose or high fructose corn syrup. And this feeds the whole fructositis kind of hypothesis, right? That fructose is causing inflammation and it's a bad thing. I do tend to subscribe to this in large part. It says one possible explanation for this paradox is the source that the elevated AGEs is coming from the intestines, not the food. So maybe the AGEs are coming from gut bacteria interactions with all this fructose that's being consumed. So again, pretty discrepant from the earlier paper that talked about how fatty meats are a bad guy, and this one's saying, fructose may be worse. So now that we're done with that shallow dive of past years, let's return to this 2023 paper that just came out. It does say existing data demonstrate the gut microbiota may be considered a source of AGEs. E. coli, for example, releases AGEs during growth, including an AGE precursor. So this is some evidence that, some of this is coming from the bad guy bacteria in your gut. It also points out that, yes, the human gut microbiota, the bacteria in your large intestines, is able to degrade Maillard reaction products. So that's another interaction. Even that it's been demonstrated that bacterial metabolites can modulate AGE toxicity, so essentially making it more toxic, worse. For example, increasing production of AGEs in the aorta, worsening aortic stiffening. So you can see there's a two-way street here. We have the diet impacting the populations of bacteria in our gut, and then we have the bacteria in our gut, particular kinds, affecting how the body creates or responds to AGEs. It's probably worth noting that changes in these gut populations, they're not completely rewritten by diet, of course, and it takes weeks. Here it says in a laboratory study, it was demonstrated that AGE-rich food significantly aggravated alterations in gut microbiota biodiversity in a time-dependent manner, being more pronounced at 18 weeks of exposure when compared to 6 or 12 weeks. This lag in how changes occur once diet changes, I think it explains quite a bit from body composition to cognitive effects and that kind of thing. You change your diet, you don't get immediate effects. Maybe it's partly because the gut bacteria need time to sort of start speaking to each other differently or repopulating or increasing their diversity. And of course, there's this concept that the good guys will generate short-chain fatty acids like butyrate that can be used as a fuel right there by intestinal cells, or acetate or propionate, things that are generally considered good things in many ways. This new paper, by the way, just to sort of wrap this up, suggests that there are some things that could be beneficial as we think about AGEs. Again, this new dietary, typically bad guy, not always. There's some fish AGEs that apparently are good, but if you're going to try to reduce these things because we know they have some connection with disease, probiotics, several studies have demonstrated modulation of gut microbiota and their association with AGE accumulation when people consume probiotics, people or lab animals. Also prebiotics, so not the actual swallowing of new bacteria, but feeding the good guys with the right fibers and whatnot. Polyphenols, also part of this. They point out quercetin that you'll see in onions or grapefruit and that kind of thing. My own research has looked at coffee polyphenols, things like chlorogenic acid, and their relationship to AGEs. Think of AGEs, at least what we might measure in your skin, as a very long-term exposure to these heat-produced gummed up proteins, or even an excess of carbohydrates like fructose, not considered a good thing. So there's a little tidbit for you, some new research and a little follow-up, as we are wont to do, on a dietary component that not everyone is aware of, but it's probably good to at least be aware that it's there, and the fact that the new thinking is that the gut microbiota are sort of this linchpin in whether or not they're good guys or bad guys and what comes of it. So thanks, everybody, for listening to this new installment of nutritionradio.org. Please check us out on YouTube, our backup, Lawnman07. Please leave a review on iTunes if you would and subscribe. But we're out of time today, so we'll catch you next time. The nutritionradio.org podcast is for informational purposes only. If you're interested in starting a diet or exercise program, check with your physician, nutritionist, or qualified exercise physiologist in order to make the progress that you need.